Hi everyone, and welcome to this class on antiviral drugs. In this class, we'll be looking at antiviral drugs, their role in fighting against virus, from common cold to more serious HIV virus. But before we dive into the specifics of this topic today, we'll be looking at what exactly are antiviral drugs and how do they work. So antiviral drugs are a type of drugs or medications, prophylactics specifically designed to combat the virus. Just like we have antibiotics, we have antibacterial. Antibiotics are generally mostly regarded as antibacterial. We also have antifungal. Then antiviral drugs are specifically against virus. Unlike antibiotics, which target bacteria, antiviral works by interfering with different stages of the viral replication cycle. And this can evolve by preventing the virus from entering healthy cells. That is, the normal cells are guided, like building a shield around the cells to avoid invasion. Or it can also act by inhibiting the viral replicating ability. That is, it prevents the virus ability to replicate its genetic material, like stopping it from making copies of the cell within the cell. Then it also act by disrupting the assembly of new viral particles. That is, peradventure the virus has been able to enter the cell, it prevents the assembly of the particle. Imagine when like dismantling all the mechanism where the virus produces its copy. So this is basically how antiviral drug works. We'll be looking at different mechanisms and so on and so forth as the lecture goes on. My name is Bamidele Odumosu. So, like I said earlier, the mechanism of the antivirus, is based on the nature of the virus it's acting on. That is, inhibiting their development rather than destroying the virus. Because it's not easy to destroy a virus. Viruses are particles. They just insert themselves in the cell and begin to replicate and causing symptoms, which is usually what causes disease. So the virus cannot be terminated, but the virus can be prevented from replicating. And this tragedy is by inhibiting their development rather than destroying the virus or preventing their critical enzymes from functioning, thereby rendering them useless and dormant in the cell. So we'll be looking at the challenges associated with production of antiviral drugs because we have more in existence, more of antibiotics in existence than antiviral drugs in comparison. So everywhere and there are easy production of antibiotics, but there have been challenges associated with production of antiviral drugs, and we will look at these challenges. Now, challenges associated with antiviral drugs are many. Now, one of the challenges is the fact that in target specificity, development of effective antiviral drugs represents a unique challenges compared to antibiotics, highlighting the complex nature of these viruses and their interaction with the host. So, we look at the key obstacles that are faced in antiviral drug production, which is first of all target specificity, and it is the fact that it is very difficult to make compounds that interfere specifically with viral activities without causing significant harm to host cells, thereby making more problem for the specific action of the virus within the cell. So, like I said earlier, the virus replicate within this host cell. So the virus host reaction rely heavily on the host machinery for replication. Therefore, it makes it difficult to target the virus without harming the normal human cells. And this specific interference, usually with virus within the cell, makes it difficult to provide target drugs which can isolate the virus without causing harm to the whole cell. For example, viral proteins often have similar structures to human protein, and this is a bit difficult in developing drugs that can specifically target the virus without harming the host. So, in specificity of antiviral drug, it's a bit challenging. Therefore, this limits the production of antiviral drugs. So we also have sorry about that. So another factor is that we have limited understanding of viral life cycle. In the laboratory, many medically important viruses such as hepatitis B, hepatitis C, papillomavirus, and so on and so forth have complicated life cycle which needs to be studied. So compared to bacteria, our understanding of viral life cycle remains incomplete, and it hinders the application of effective drugs which can be designed to interrupt or disrupt this replication. So it is difficult to understand replication, and in the laboratory, these studies are limited, hence making it a bit challenging to develop drugs that can, that can be used to study their life cycle. Because if you don't understand the life cycle of a particular pathogen or understand how this pathogen grow, how can we limit the growth, how can we prevent the growth, it becomes a bit challenging. So the laboratory study and limited understanding of viral life cycle is a challenge in making antiviral drugs. Then another factor is the fact that there is a rapid mutation by virus. Viruses mutate frequently and allow them to develop resistance to antiviral drugs and render them ineffective over time. And these necessitate constant development of new drugs and strategy to stay ahead of viral evolution. So their fast mutation makes them makes it a bit more difficult to produce drugs, and they remain effective. They remain ineffective for a longer time. And if you compare it to the rate at which bacteria undergo mutation, virus go faster. They are more faster in their mutation than than bacteria. And also, we also have a wide variety of viruses. There are so many different types of viruses, and they all have their own unique characteristics, making it more challenging, and it's a bit difficult to develop a broad spectrum antiviral drugs against that of a drug such as antibiotics, broad spectrum activity like ciprofloxacin that can kill both gram-positive and gram-negative bacteria. But for the type of variety of virus we have, they have their own unique characteristics. They are, the way they grow and the enzyme they require for their growth are a bit diverse and unique. So it's a bit challenging to develop such drugs that can actively kill a broad range of virus. Then also we have a high cost of development, in the sense that there's a lengthy and complex process of antiviral drug development coupled with the high failure rates and the potential for limited market size. This creates a significant disincentive for pharmaceutical companies to invest in this area, because they are out to make profit. They are not interested in trials and losses and failure. So they get discouraged in this. They want to invest in drugs that can easily be marketable and profitable. So the cost of development of antiviral drug is huge, and most companies are not interested in investing in this area. Then also, human trials resulting in antiviral drugs can also be difficult. Testing drugs can be difficult, and it's very expensive, because most people know that once the virus infects cells, the virus remains there for a very long time, will be dormant and so on. So what if the virus is going to have a very, let me say, the very the toxic disadvantage or more uh, mortality or morbidity rate in the system. So in testing for such drug with human trials, it's a bit challenging. Nobody wants to volunteer to submit him or herself for this trial, which is, don't know the outcome. So this also hinders development of antiviral drug, because once a drug is developed, there's a need for human trials to ascertain the effectiveness of the drug as quality control, to determine if this is the adverse effect, this is the most potent way, this is how the drug delivers in the body. So there are less volunteer for this human trial, even though there's a huge lot of money that's being given. Then also have poor delivery of antiviral drugs, in the sense that antiviral drugs need to be able to reach the drug target and prevent replication of virus in time. But the problem with this is that there are many drugs, many antiviral drugs are being destroyed before it gets to their target, and gives a poor delivery method in terms of drug delivery system. So the future and current advancement in antiviral drug, based on the fact that we've seen all these problems and all that. So what is the way forward? What is the way forward? It is not as if there are no drugs that are being produced, but the fact that there's a need for revolutionizing the way at which antiviral drugs are produced. So one of the ways is the RNA interference, which is called RNAi. It's a method that can be used to silence genes. It is an interesting natural process in cells that allows them to silence specific genes. It's like a built-in volume control for gene expression. They reduce, you can reduce the volume of the gene activity, gene expression, or can tone it down and increase. It's like a silencing. It holds an immense potential in fighting virus, because they rely on replicating their cell, their RNA within the whole cell. So this technology can be used in several ways for antivirus production, whereby scientists can design small interfering RNAs that are called siRNAs, specific to essential viral genes. These small interfering RNA that is called siRNAs, once delivered into the infected cell, can actually silence the targeted genes, which by hinder their replication and also hinder their spread. Now, this approach has been shown to be promising against various viruses such as HIV, hepatitis C, and influenza virus, which are one of the most prominent and prevailing viral infection. So it has been shown, it has been demonstrated to show promising activity against them. So we also have gene editing. Gene editing is actually a technology that has been known, used in editing genes. Not only virus, can be used for bacteria and so on. So the technology is known like a genome editing. It's a powerful technique that is used to modify organism DNA at specific locations. It's like searching, finding, and editing specific codon within a vast number of genes. Once this is found, once this is detected, then the particular gene can now be deleted or altered, to change specific bit of the genetic information that is being coded on the sequence. Once this is done, then the particular gene that is for the infection is now altered and make the virus to be less effective or non-effective. It's like editing the gene. The gene that codes for a particular violent gene is edited to make it non-violent. Simple as that. And in the fight against virus, researchers are exploring numerous ways to utilize gene editing for this antiviral purpose. And the benefit of gene editing for antiviral is like it's specific. That is, it is targeting specific harmful gene that is going to be, toxic to the human cells, and it's targeting them. Then also it is a bit more doable in terms of how can this gene be, how can this gene be modified and be permanent? It's a bit durable that offer a long-term protection, and those activity does not really are affected by mutations, and so it works against a broad application like bacteria, viral, fungal, any type of infectious gene. It's a bit more stable and applicable against virus and potential future mutation with viral cells. So we are looking at now types of antiviral drugs. In the types of antiviral drugs, we have nucleoside analogues. Nucleoside analogues were the first compound that are shown to be effective against viral infection. A nucleoside analog is a synthetic drug molecule that closely resembles the natural viral nucleoside, which is a building block of DNA and RNA of virus. They have slight modification to their structures, which allow them to actually mimic or appear similar, or actually mimics the virus cell and interfere with the normal process of virus replication. They have a structure that looks like the nucleoside. Example is the guanosine and 2-deoxythymidine, and they act by interfering with the synthesis of this viral nucleic acid. So they actually act as a competitive inhibitor during virus replication. Example of this is clover. It's a drug that looks similar to natural nucleoside, and they can be incorporated in the viral DNA or RNA chain during replication. Because of their altered structure, it will not allow the chain to continue, that is, the sequence, and they disrupt the virus' ability to reproduce. And this drug, clover, acts by inhibiting the replication of herpes virus by targeting the DNA polymerase. It is one of the examples. There are so many other ones that actually work with this mechanism of action and specific viral with a target. This is the mechanism of action of the drug. You can see how this process goes on. It acts by inserting itself into the sequence, and once it binds to the viral polymerase, it prevents it from continuation, and once it prevents it from continuation, it terminates the sequence of the replication process, thereby making the virus to be dormant. This is a diagram that was described. I'm just using this as an example for our easy understanding of the process. We also have antiviral drugs for treating HIV/AIDS, which is called HAT. Now, we have those number 1, 2, 3, and 4. Number 1 is the NRTIs and the NNRTIs. Now, they are both medication, they are both drugs that are used in antiretroviral therapy, which is the ART, used in treating HIV/AIDS. The NRTIs is nucleoside reverse transcriptase inhibitors or nucleotide reverse transcriptase inhibitors, NRTIs. They work by targeting different parts of the viral life cycle, but they ultimately interfere with HIV ability to replicate. We also have the NNRTIS, which is the non-nucleoside reverse transcriptase inhibitors. Now, the NRTIS, which is the nucleoside reverse transcriptase inhibitors, work by mimicking naturally occurring building blocks of DNA and RNA, just like we have the other one. But once it's inside this host cell, the NRTIs are mistaken for the real building blocks, and they are incorporated into the HIV growing DNA chains. And these will cause errors or cause mutation and prevent the virus from replicating properly. So example of this is we have the lamivudine, tenofovir. We have so many of them. For the non-nucleoside reverse transcriptase, we will discuss that one in details. As we look at other ones, we have the protease inhibitors, we have the fission inhibitors, and have the integrase inhibitors. So, in looking at the non-nucleoside reverse transcriptase inhibitors, they actually act by non-incorporation into viral DNA. They exhibit direct binding, non-competitive to reverse transcriptase enzyme. This enzyme is actually responsible for the replication of the virus. It's the one of the most popular target in the field of antiretroviral drug development. An example include the nevirapine and delaviradine. They are examples of such antiviral drugs. They don't mimic building blocks, but they bind directly to specific pocket on the reverse transcriptase enzyme itself, and it prevents the virus from properly converting HIV RNAs into DNA, halting its replication. So the challenge associated with the NNRT is the issue of drug resistance, because the virus can actually replicate in the presence of these antiretroviral drugs, and this normally occurs when there is single therapy, that is monotherapy, when the drug is used alone. So to prevent this, there is usually a combination therapy, which is called the HART, the highly active antiretroviral therapy, which prevents replication in presence of this drug. That is, drugs are combined in combination therapy that allow the drugs to attack from different angles on different mechanism of action. Some can actually act by preventing a particular enzyme or particular mechanism, so it will not allow the drug to replicate in the presence of more or two or more drugs. So the treatment regimen usually combine 3 drugs, at least 2 different classes of antiretroviral drugs, to prevent resistance. That this is the only way at which this challenge can be resolved. So the advantage of this drug is that it's highly effective. It suppresses HIV viral load. When the viral load is suppressed, then the person feels okay, because it prevents replication. Once a viral replication can be contained, then the individual that is being infected can behave normal, because the virus is no longer replicating, and it also have fewer side effects. They are usually generally tolerated by cells. They don't show more they don't show more side effects compared to other antiretroviral drugs. That is, the non-nucleoside reverse transcriptase inhibitors don't show more side effects. They show lesser side effects than other antiretroviral drugs that do not use this mechanism. And it is also a once daily dosing, that is, it can be taken once daily, and this can actually be convenient rather than taking 3 times daily, because in the management of viral infections such as HIV, some patients have to take 3 drugs daily. I mean 3 dosage, morning, afternoon, night. But in this case, it can be once daily, which is a bit more convenient. Among the disadvantages, apart from the challenges, is that HIV can develop resistance to this drug. Like I said, that's one of the challenges. But now we're looking at it under the disadvantage. HIV can develop resistance to it quickly. And also there is potential for drug interaction. That is, the non-nucleoside viral transcriptase inhibitors can interact with other medications. And there is incompatibility with some drugs if the patient is, because when people with HIV when they are being treated, sometimes they can be open to other infections. That's why they call it syndrome, because it can open up to other infections. And when these infections are being treated, there could be incompatibility with these drugs. So there could be drug interaction. So this is one of the side effects, and it's also neurological side effects, whereby some of these drugs are actually called dizziness and insomnia, that is a lack of ability to sleep, and also nightmares. So we also have another type of antiviral drug, which are the protease inhibitors. Now, protease inhibitors are specific type of drug that block the activity of viral enzyme called protease. Protease itself play a crucial role in the life cycle of various viruses, which include HIV, hepatitis C virus, SARS-CoV-2, and so on. So by inhibiting protease, this drug can interfere with the virus' ability to mature, replicate, and spread within the cell, and effectively slow down the progression of the virus in the cells, thereby improving the patient overall health and so on. So the protease inhibitor prevents viral replication by selectively binding to viral proteins, e.g. HIV-1 protease. They act by preventing the virus from producing new copies of the cell and effectively slowing down or stopping the infection. And they act in specific ways. They have the competitive, non-competitive inhibition, and transition state inhibition. So let's look at this competitive, non-competitive, and transition state inhibition one after the other. So for the competitive inhibitor, the inhibitor molecule exhibits competitive binding to the active site of the protease enzyme, effectively mimicking the natural substrate and consequently impeding the processing of viral protein by the target enzymes. So if you look at the figure here, the diagram here, it shows the competitive inhibiting action of this drug, which prevents the natural substrate and inserting itself into the viral cell, thereby preventing the viral protein from being processing. For the non-competitive inhibition, the inhibitor molecule binds to a different site on the protease enzyme called the allosteric sites, thereby inducing conformational changes in the enzyme. Now, allosteric sites are special locations of proteins, particularly enzymes. They are distinct from the active site where the main function of the protein takes place. These sites act as regulatory switches. They are like a switch. They allow molecules called effectors to bind and influence the protein activity in various ways. So, by binding to these allosteric sites, it makes them inactive and prevent them from reaction. So, if you look at the diagram, this is the enzyme, this is the substrate, and this is the inhibitor. Once you bind to it, you switch it off. You switch it off, and there's no activity again. So the transition state inhibition This is a very different method of uh, action, mechanism of action. Now, imagine an enzyme like a protein having a lock and key substrate in quotes uh, reaction. So when the substrate fits into the enzyme active site, they form a complex. Fine. But in the transition state, this this complex is catalyzed by the reaction. The complex is catalyzed by the enzyme, and it doesn't happen instantly. It has to reach a high energy threshold, and this high energy threshold gives an unstable intermediate state called the transition state, where the substrate is distorted and partially converted into the product desired by the enzyme, by the viral cells. So now, what this inhibitor does is that they bind to the molecule and they can mimic the transition state. When they now bind to the active site, but they will not do this in the same way as the substrate is expected to do. Instead, they fit like a mismatched key that gets stuck, and it will, rather than going to a higher state, it will stabilize it. By binding to the transition state, the inhibitor essentially holds it in place. Instead of getting to more stability, it makes it more stable. And this stability is not good for the reaction because it has to react, it has to go in an unstable state. The unstable transition state is what allows the reaction to proceed quickly. So by stabilizing it, it slows it down. By slowing it down, it prevents it from acting. So the inhibitor occupies the active site, preventing the actual substrate from binding and completing the reaction. So it will prevent the reaction. So as a result, the inhibition blocks, in quotes, blocks the enzyme from preventing it from converting the substrate into the product. So this is actually one of the mechanisms of action that prevents the virus, and it is usually by protease inhibitors. So we also have fusion inhibitors. Fusion inhibitors are specific type of antiviral drugs that prevent virus from entering the host cell. They achieve this by interfering with the fusion process, which is the critical step where the viral envelope merges with the host cell membrane. And this allows the virus to deliver its incorporate its genetic materials and establish an infection within the host cell. So, by targeting viral envelope, many viruses include HIV, influenza, Ebola, and so on. They have an outer envelope made of protein and lipids, which play a crucial role in their fission process. So what the fission inhibitor does is specifically it binds to these viral envelope proteins, particularly those involved in binding to host cell receptors at the outer cell, and it mediates their membrane fission. So the binding blocks the crucial conformational changes required for the virus to merge within the cell, thereby leading to non-infectivity. So currently, the only licensed fission inhibitor for HIV treatment is efluvitide, which is a freezer. It is a synthetic peptide that mimics a region of the HIV envelope protein GP41. Binding to GP41, efluvitides prevent the conformational changes needed for viral fusion within the cell membrane. So we are we have the mechanism of action. It blocks the fusion of viral host cell membrane. Now, what are the advantages? The advantage is that it is very potent against HIV. That's why we are having decrease in HIV infection here and there because some of these new mechanism of action, new antiviral drugs have been effective, especially it prevents those molecular resistant strains. It is well tolerated with less side effects. It has its own unique mechanism of action, making them a valuable option for patients who have failed or who have failed treatments as regards antiretroviral therapy. So the advantage is that it requires injection administration, which is not convenient for some patients. It is also expensive, and sometimes it could cause reaction at the injection site. So these are the disadvantages of this drug. So, but I believe that there is ongoing research into how this can be delivered without being used as injection administration. So now the next one is second to the last is the integrase inhibitors. The integrase inhibitors are a specific class of antiviral drugs that is used primarily against HIV. They work by targeting crucial enzyme called integrase, which plays a vital role in the viral life cycle. So what is an integrase itself? Integrase is an enzyme produced by the virus during HIV infection. So the virus creates a DNA copy of its RNA genome, and the integrase play a critical role in integrating this viral DNA into the host cell DNA, like hijacking the cellular machinery to replicate the virus and continue the infection. So by blocking the integrase, the viral DNA remains unintegrated and unable to express its gene, and automatically it halts viral replication and prevent the spread of infection within the host cell. So this is the way uh, integrase inhibitor works, and it has its own advantage. It is it is highly potent, effective at suppressing HIV viral load. Again, once HIV viral load has been suppressed, then the person feels better. Then it's durable in its efficacy. It has a high genetic barrier to resistance, and it also has good tolerability. It's well tolerated within the cells, and it has fewer side effects compared to other antiretroviral drugs. And the dosage is convenient. It's also available as once daily medication. The mechanism of action of integrase is bind directly to HIV integrase enzyme, preventing it from performing its essential function like AppSec, and it prevents the viral replication. And it does not have any human homolog cell. That is, it does not have any equivalent counterpart in the human cell, making it highly selective for virus and reducing the risk of side effects. So because if you look at the beginning, we talked about the challenges associated with providing more antiviral drug, and one of the challenges is that specificity of the antiviral drug, because when the viral integrates itself, it becomes difficult. But this time, this drug has no human homolog. That means once it's applied, it goes straight for kill. It goes straight to the virus. Wherever the virus is hiding within the cell, it goes straight and selectively, you know, bind to it. The last but not the least is among our interferons. This very popular uh, antiviral components. Interferons, they are natural signaling molecules produced by human cells. They are like a first line of defense against vast variety of, vast infection, bacterial infection, viral infection, and so on. They are like the alarm bells that alert nearby cells about the intruder, antigens, and so on. And they trigger a kind of a multi-pronged antiviral responses. So they belong to the class of glycoprotein known as the cytokines that they naturally possess antiviral activity. Not only antiviral activity, they also possess antibacterial activities. So once they are released by the host cell, like a bell, once they give like a signal, they are released by the host cell in response to the presence of the antigen or pathogen such as virus, bacterial, parasite, or tumor cells. And the way they act is that they they are like a signal, like I said. They detect the virus, they send the signals, and they activate antiviral defense. And they bind in a signaling cascade manner inside the cell, leading to the production of hundreds of antiviral protein encoded by interferon stimulated genes. So they are named for their ability to interfere with viral replication. So that's what we call it, interferon. They interfere and they protect the cell from virus replication. So let's look at types of interferons. Oh, sorry, let's look at the function. So they activate immune cell, I've said it before, such as natural killer cells and macrophages. They also increase host defense by upregulating the antigen. They make the antigen more presentable, more palatable, more, they present it for the antibodies to actually bind to it. We have type 1 interferons, which are also known as the IFN or leukocyte. We also have the IFN fibroblast, and we have the uh, type 2, which is also known as immune IFN gamma. They are examples of interferons and types that are found in the cell. So they are natural activating, viral deactivating molecules within the human cells. Thank you for listening. Some of the credit is shown below. Thank you for listening.